Pnawn da, a chroeso cynnes I chi gyd. Well, good afternoon and welcome everybody. It's great to have our alumni, staff, donors, friends and supporters here today, and thanks to all of you for joining us. You've just heard that this digital showcase is being recorded, and we'll send you the link after the event, and you're welcome to share that. I hope you enjoy it. So for those of you that don't know me, I'm Professor Colin Reardon. I'm President and Vice Chancellor of Cardiff University. And we've invited you here tonight as part of the Cardiff community, comprising graduates who finish their studies at Cardiff and also friends who are kindly supporting the learning research of the university, and you're very much valued. Thank you. Now, you may know that October is Breast Cancer Awareness Month. So this evening, our focus is on the work of the European Cancer Stem Cell Research Institute, or ESCRI, looking at the research the team is doing into cancer stem cells in breast cancer and their role in metastasis, the spread of malignant tumours around the body. So crucial stuff here tonight, and I'm sure it's going to be an interesting hour or so. I'm delighted to be able to introduce two of our colleagues working, of course, in this area. That's Professor Richard Clarkson and Dr Giusy Tornillo. Richard's the Deputy Director of ESCRI, and his lab focuses on identifying new ways of preventing tumours from spreading around the body, and the utility and importance of that is, I think, very apparent. And also understanding the changes that occur within tumour cells that cause them to metastasize and spread. They're also working on the development of new therapeutic strategies, new treatments, either to prevent metastasis from taking place or to eliminate the cells altogether. Dr. Giusy Tornillo works in the same lab, and her research looks at how to understand the molecular mechanisms, what's really going on at a molecular level in the regulation of normal tissue homeostasis, and how cancer cells hijack these mechanisms in order to fuel their growth and aggressive behaviour. So it really does, how does the cancer take hold of normal healthy tissues and take them over and begin its own uncontrolled growth. So we're going to be hearing from both of them, of course. That's the main event this evening. In terms of the format, what we'll do is we'll hear from our two speakers tonight, Professor Clarkson and Dr Tornillo, and then there'll be an opportunity for a question and answer session at the end of the presentations. So if you do have any questions as we go along, or at the end, indeed, of the presentations, just enter them into the Q&A box at the bottom of your screen, or wherever it is on your screen, or mine it's at the top, at any point, and we'll get to as many questions as time allows. And if we don't get round to your question, I'm sure it'll be possible for us to give you a reply subsequently. So I'd like to start then by welcoming Professor Richard Clarkson, who's going to give our first presentation. Richard, please.
Thank you, Colin. And while I'm just getting this screen up and active, I just want to also welcome you all to this virtual visit to the European Cancer Stem Cell Research Institute, of which I'm Deputy Director. So hopefully now you should be able to see the slide in front of you. And so we can kick off. So yes, so the European Cancer Stem Cell Research Institute, well, I'm very pleased to say that actually we're now back up and running within the labs. It's been a difficult time during COVID, of course, and there's been really a great deal of work going on behind the scenes at Cardiff University to get all the researchers back into the research labs as quickly as possible. And I'm very delighted to say that the 60 or so staff and students, PhD students, are now back in the labs. We're working a shift model, but we're all back up and running, and cancer research is going ahead as we'd all hoped it would have done. So we're very pleased about that. So as Colin's pointed out, Giusy and I are going to give you a quick run through of a couple of projects that are going on in my lab and in her lab. We're both cancer researchers in our own right, and we both specialise in breast cancer. So we thought we'd give you a flavour of the projects going on there. But to start with, I thought I'd spend a couple of minutes just giving you an overview of what a cancer stem cell is and why we would have built an institute around that concept, and why we think it's such an important area of biology and cancer research, with the potential, hopefully with the potential for advancing new therapeutics in cancer. So what is a cancer stem cell? Well, I think the best analogy that I can come up with that helps to explain what a cancer stem cell is if we liken a solid tumour within a patient to a tree. And it just so happens I have a tree here in front of me. This is one of the trees in front of the Hayden Ellis building where the institute is based. If we wanted to get rid of one of these trees, maybe to give us a better view of the building, perhaps, I don't know, we'd come along with a saw and we'd cut this tree down, and then we'd treat the roots with a chemical toxin to make sure that we destroyed the roots of the tree as well. Well, that's basically very similar to what we do in the clinic. If a tumour is accessible to the surgeon, it's very likely that a surgeon will remove the tumour from its site, and then it's likely also that the patient will undergo some form of chemotherapy afterwards to try and poison the roots of that tumour to ensure that, to the best of our ability, those tumours don't come back. But the problem is, of course, is too often and with some prevalence, patients do find that eventually those tumours do come back. And it's that problem that's a really important clinical unmet need of trying to prevent those tumours from returning, which is at the heart of what the Institute does. So the analogy with trees doesn't just stop there. So what we find is that if you look at a tree, of course, you can see the trees and the branches, and it's made up of very different structures. But what you don't see are the stem cells or the seeds in those trees. The seeds, of course, are very small and they're very rare. Compared to the rest of the tree, the bulk of the tree, the seeds make up a very, very small component. But of course, they're the most important part of the tree. They're the part that will disseminate new tree growth in the local environment and may well get caught on the wind and get disseminated across Cardiff. And that again is very much like we now envisage a tumour within a patient. A tumour is made up of very different cells, just like you see the branches and the trunk and the leaves, etc. on a tree. It's made up of different structures, but it also contains seeds. And these are the cancer stem cells that we now describe within the Institute, and others have recognised around the world now. And just like on trees, the seeds or cancer stem cells within a tumour are very rare. And it's these very rare cells that are responsible for some of the most important clinical problems that we see. What are those problems? Why are they important? Well, we now recognise that it's these very rare cells that are responsible for the cancer spread or metastasis of the tumours around the body. Actually, the bulk of the tumour, 99% of that bulk of the tumour, is incapable of forming new tumours elsewhere in the body. And it's less than 1% of those tumours, in most cases, that are likely to be responsible for that. The other characteristic of a stem cell within a tumour is it's very resistant to the chemotherapy. So it's a double whammy, if you like. You have these highly resistant cells, very rare, but it's only those cells that are responsible for the dissemination of the tumours elsewhere. So what does that mean? So if what we need to do is understand the biology of cancer stem cells, and that's where we come in. We've recruited a number of eminent researchers within the Institute now, and we're starting to understand the biology of cancer stem cells. As I've shown here, this is a tumour that from my lab that we've managed to grow within the laboratory. And we've labelled up the cells within this tumour, just labelling up the cancer stem cells. And as you can see, it's only a few of these cells within this bulk of tumour mass that actually have cancer stem cell properties. So by labeling these, now we can start to understand the mechanism of action. What makes them different from the other cells within that tumor bulk? If we can understand the biology of cancer stem cells, then perhaps we can identify new therapies that target cancer stem cells. And by targeting this tiny proportion of the tumor, we're, if you like, turning therapy on its head. Rather than focusing on debulking the tumor, we're getting at the heart of how that tumor then disseminates around the body and causes so many problems. So I'm going to turn my attention now to the specific project in my lab that involves breast cancer, and why, this particular project has a particular relevance to the cancer stem cells. So this project started quite a while ago now, 2012, and is really the culmination of 3 consecutive PhD projects within the lab. My first PhD student on this project was Luke Piggott, and he identified this molecule called c-flip within cancer cells that he showed was responsible for a decision-making process within the cancer cell. And that decision-making process was stimulated by the immune system, by the immune cells of the patient. What we now know is that immune cells are extremely good at getting rid of cancer cells. They're very, very good at it, and there are many therapies out there now that are trying to enhance that immunity against cancer. And one of the mechanisms by which the immune cells destroy cancer cells is by stimulating this receptor on the surface of a cancer cell called trail. Now, the problem with cancers that become resistant to the immune system and therefore start to form for long periods of time in a patient is that this pathway becomes resistant. And the reason it's become resistant, Luke found, was that this molecule, c-flip, is very highly expressed in cancer cells. And what it does is it swamps the cell with c-flip, and it starts to attach itself to the receptor of the trail and switches off the ability of the cell to initiate a cell suicide that is instructed by the immune cell. So what you basically have now is the cell's inability to undergo a cell suicide, and this c-flip molecule being responsible for that. So this is where my second PhD student, Liv, came into play. She was recruited along with my collaborators, Andrea Brancali and Andrew Westwell from the School of Pharmacy in the Cardiff University. We worked closely with them, and what they did, what Liv did under their supervision, was to use computer modeling to identify how this c-flip protein attached to this receptor. So that interface there, where those 2 proteins interact, is what the computer modeled here. And they looked at the shape of this pocket that was responsible for this interaction and tried to design a drug, a new drug that would, so a small chemical that could fit into that pocket like a key and lock and stop the interaction between the receptor, here termed FAD, and the c-flip molecule itself. The key to this working in the clinic would be if this chemical compound was not able to inhibit the ability of the important caspase 8 molecule from binding to the same receptor, because it's this molecule here that's responsible for the cancer cell death. So what we had to find was a chemical that would not inhibit that interaction, which is here, but would inhibit this interaction here. And I wouldn't be sitting here telling you about this if we hadn't found one, and we did. So Liv's successful PhD was to find a drug, a small compound called OH14 that binds into the pocket of c-flip and prevents c-flip now from inhibiting this pathway and killing the cancer cells. And what she found from Luke's work was that cancer stem cells are exquisitely sensitive to OH14. If you add OH14 to cancer stem cells, these cells become very sensitive again to this trail-mediated kill. Tim Robinson then came into the lab. Tim was a clinical PhD student. He's now a senior oncologist at Bristol University. And he graduated from my lab in 2018, but his project was specifically to take this new agent of ours, which is a completely novel compound, and apply it to triple-negative breast cancer. And his particular interest as an oncologist was that he was interested in the relationship between chemotherapy and the relapse of tumours following chemotherapy in triple-negative breast cancer. So triple-negative breast cancer patients, it's a more aggressive form of the disease, and very often patients will relapse. The tumours will come back after chemotherapy, and Tim wanted to understand that process. So Tim took a number of triple-negative breast cancer tumours and he treated them with paclitaxel. And as we expected, the paclitaxel debulked the tumour, so eliminated most of the bulk of the tumour. But the surprising finding from his work was that the influence of paclitaxel in destroying the bulk of the tumour was actually opposite in the stem cells. The stem cells of the tumour were proliferated. We had more stem cells after paclitaxel treatment in their treated tumours than we did originally in the untreated. So this suggests that there's a strange relationship between the chemotherapy and the stem cell. So the theory behind his next piece of work was to say, well, maybe we can make these very sensitive to the immune-mediated killing of the immune cell through trail. And the way that we could do that is to use our agent OH14. So what he did is he combined paclitaxel with the OH14 to see if he could now target the stem cells. And that's what he found, with really quite striking effect. So this data here, this is the last of my data that I'm going to show you now, is an illustration of an experiment where he took human breast cancer cells and he put them into a mouse, and the tumours grew inside the mice very rapidly if the mice were untreated. If we treated the mice with paclitaxel, as expected, the paclitaxel was excellent at debulking the tumour, and we were able to regress these tumours right down to virtually undetectable levels, just as you would see in the clinic. However, what Tim did is he watched and waited, and after about 8 weeks, he found that the paclitaxel treated animals, the tumours started to return, and they started growing just as fast as the original tumours did. So what he did is he combined paclitaxel with our agent OH14, here shown in blue. And just as before, the tumours through the paclitaxel regressed, but now the tumours stayed away. And this is the effect of the OH14. So what it appears to be is happening here is that the addition of the OH14 with paclitaxel removes the stem cells as well, and now these tumours don't return. So the consequence of this, it will be a manuscript that we're now preparing for 2020 because of COVID got in the way. We'll be publishing this data, and hopefully in the future we'll be trying to apply this to clinical trials. I'm going to pass over to Giusy now. She'll explain her project and I'll come back to you in a second.
Thanks, Richard, and thanks to all the organizers for having me here today. So my name is Giusy and I have been working as postdoc in Professor Smalley's lab at Cardiff University for about 7 years now. And today I'd like to share some of our work with you. So my research focuses on understanding more about a protein called llyn, which we believe can be a very good target for drugs against breast cancer, and specifically against those cancer stem cells which, as nicely described by Richard, are able to spread around the body and form metastases. And one of the things we have learned so far is that in some cases, it is better to stop plan A and switch to plan B. And hopefully, by the end of this presentation, you will understand exactly why and exactly what I mean with this. So a few more details about llyn. Llyn is a protein present in almost all the cells of our body, and its function is to transmit signals. For instance, in response to specific hormones or growth factors, llyn can turn on or off other proteins, which can make cells grow or move. And one very important piece of information to keep in mind is that breast cancer cells have much more of this protein, much more llyn than normal, non-cancerous cells. And to be precise, there are two versions of llyn. One is called llyn A, and the other one is called llyn B. And as shown in the schematic here, they actually look quite similar to each other. And if you think of a protein as a string of bits, limb B is missing only a few bits at the beginning of its string. But do limb A and limb B play the same role, or do they play different roles in breast cancer? And no one before us asked this question, but now we have an answer for it. So we went and measured the amount of limb A and limb B in a number of breast cancer, human breast cancer samples, as many as those little dots you can see in the graph on the left. And what we notice is that some of those cancers had a very high limb A, limb B dose ratio. In other words, some of those cancers had a lot of limb A, but only a little limb B. And when we went to compare the prognosis of this cancer to the rest, we found that breast cancer with high limb A and low limb B had the worst prognosis. This means that patients with breast cancer with a lot of limb A but only little limb B, unfortunately, do survive less long. So we started a series of experiments in vitro to try to understand whether limb might contribute to the aggressive behavior of breast cancer cells. And if so, which limb, A or B, might contribute to it. So we genetically manipulated breast cancer cells in vitro to generate cells with no limb at all, with only limb A, or with only limb B. And we then compare the ability of all these cells to migrate and invade in vitro. So cells with no limb at all, just like cells with only limb B, could not migrate or invade very well, whilst cells with only limb A could migrate and invade very effectively. So this is one piece of evidence that breast cancer cells employ, use, and need limb A, but not limb B, to be aggressive. So next, we thought, since, if you can, next slide, please. Given that limb A seems to be the bad guy and limb B seems to be the okay guy, why don't we try to turn limb A into limb B in the attempt to tame somehow breast cancer cells? Of course, by using an approach other than a genetic manipulation, but an approach which could be feasibly translated into a therapeutic approach. So we designed a molecule with the ability to interfere with the production of limb inside breast cancer cells. A molecule which can make cells switch from producing limb A to producing a limb B. And here in the graph on the left, you might see the amount of limb B in purple and limb A in green after or before the treatment with such a molecule. And as a result of treatment with this molecule, cells could indeed migrate less well. So of course, we're really excited about these preliminary results in vitro, about the fact that we might be able to tackle one of the major and probably the most threatening trait of breast cancer stem cells. And of course, we'd like to test the same approach in vivo in mice soon. So thanks for listening, and I will leave Professor Clarkson wrap up and say thanks to all of you again.
Thank you, Giusy. So you've had very, very brief introduction or summary of a couple of projects in the lab involving breast cancer, both of which have identified some fundamental mechanisms that underpin how cancer stem cells work and why and how they end up migrating and getting into the tissues elsewhere in the body. And by identifying how they work and understanding those mechanisms, we're now starting to think about ways of targeting those. So I'm just going to finish now just by acknowledging the support of our funders for these two pieces of work particularly, and other research that goes on within the Institute. So I must acknowledge Breast Cancer Now, the charities Breast Cancer Now and Breast Cancer Research Aid, but most importantly the public donations. So we do receive a number of public donations, through the university, and they go straight into the universities, into the institute, and are directly siphoned on into research at the lab bench. So all of the donations go directly to our PhD programs and to our researchers at the bench, and we're very grateful for those. Thank you. So I'm going to now pass back to Colin. So thank you very much for listening, and I'll be very happy to take any questions.
Well, thank you very much indeed, both of you. I must say I did find that fascinating, and you know, we're all at the moment constantly watching things about receptors and so on and so forth, and it's interesting to see things that echo what we hear about coronavirus all the time in rather a different way. So we do have some questions. We got a couple of quite I would say perhaps more technical ones in the Q&A, which we will come to, but we've had some in advance, and one or two of those are more sort of, as it were, general treatment questions. And I'm afraid I'm going to put to you the one which you'll get all the time, and there won't be any very clear answer probably. But Marjorie wants to know, can stem cell treatment cure cancer?
Well, that's the hope and the expectation, but we need to define what we mean by stem cell treatments. So most people will recognize that stem cells are parts of all of our bodies, that we have them in the brain, we have them everywhere, and we regard those as normal stem cells. And they're essential for normal tissue homeostasis, in other words, for the normal functioning of the body. What we're talking about here is a really very different species, a very different fish altogether. These so-called stem cells within cancers are derived within the cancers, the tumours themselves. So we're talking about cancer cells that have the properties of stem cells, but they're not actually, it's very unlikely that they really resemble the normal stem cells in our bodies. So that's just one important distinction to make. If we are thinking about therapies, we want to be targeting these cancer stem cells within tumours. But stem cell therapies that you might hear about in terms of Parkinson's disease, etc., they're going to be very different approaches to targeting normal stem cells, or stem cells that may need stimulating in the brain. But that's the expectation and the hope. And as hopefully you would have appreciated from at least from our talks, is that we're starting to identify new therapeutics now, completely novel therapeutics that could target some of the functions of these stem cells that we're starting to elucidate.
Thanks very much. And it was very clear, showing which cells are the ones that cause the spread, and your method for targeting them. There's an interesting question in the Q&A, the first one that came in from Miriam Cotter there. Why the novel molecule route rather than repurposing existing?
So we do. Repurposing is absolutely essential, and it certainly saves you a lot of pain and time along the drug development pathway. So there are a number of projects going on in the lab looking at plant extract products, existing chemotherapies, existing targeted therapies, and using them in combination is absolutely key. So we do have a number of projects that are looking at that, and are with relative success, actually. So it's sad that I can't tell you about every project going on in the Institute, but it's certainly on our agenda, yeah.
Great, thanks. And another, I would say, slightly more technical one, Justin. If the chemo drug, well, can you read it yourself there? Plus FLIP-inhib co-administered, I think this is jargon. Does the chemo not inhibit immune function too? Especially
yeah, that's a very good question. Very good question. Yes, it does. It doesn't completely inhibit it. So there's a kind of there's a two-pronged answer to that. One is that we don't think that the cells that would deliver trail would be necessarily impaired by the chemotherapy to the extent that it wouldn't work. Secondly, that you very rightly pointed out a rather oversimplification in my presentation. It's difficult to get all the nuances across. The immune cell-mediated killing of the cells via trail doesn't have to be provided by immune cells either. You can actually provide trail, stimulate the cells to die through trail without any use of the immune cells. However, I just wanted to highlight the fact that it is a normal, natural process of the immune system to use this sort of killing mechanism. So one of the big areas in cancer research at the moment, of course, is immune therapy. And if we can perhaps join forces with the immune therapy, where if we can enhance the immune system and make the cells much more sensitive to these immune cells, once we've activated the immune system, you get a double effect, really. You get an amplification of the result.
I'll just say you're both doing a great job of, or have done a great job of making it accessible to a general audience as well, but there's bound to be nuances, therefore, that need to be further explained. And Byddig, whether your work's transferable to other cancers, bowel or blood?
I'll speak for myself, and then I'll pass over to Giusy about her project. But yes, so the OH14 project, we've looked in a number of different tumour types at the moment, and we're seeing successes in pancreatic and colorectal models, as well as our breast cancer models. We've not looked in any others at the moment.
Regarding lean, which is the product we are interested in, I can say that there are other solid cancer where lean is a target of interest, or at least a potential target of interest, such as I can think of prostate, pancreatic, and also glioblastoma. And it has been already well studied and somehow targeted also in leukemia. The novelty of this study, the one we're doing in breast cancer, is looking at, again, the two lean forms, and now actually we can target this aspect to try to, let's say, switch off the ability of breast cancer cell to migrate. And this is something that has not been done yet in other cancer, just because these data are quite novel. We published this data less than around 1 year ago. So I expect other people might be interested to study this aspect in other cancer.
So some exciting prospects there for the future. Raisha wants to know, have you observed a difference between CSC and triple negative breast cancer in comparison to other forms such as estrogen positive? Not sure who that is. Yeah, go ahead.
Yeah.
No, the only thing I can say that we didn't do ourselves, but there are some studies available out there suggesting that triple negative breast cancers are enriched in these cancer stem cells. So the limitation of the study is that different people did use different ways to identify cancer stem cells. So it is not easy to actually compare very often results from different laboratories. But this is the communal, let's say, opinion that, again, triple negative breast cancers might harbor a higher proportion of them. That's why they are also more aggressive.
Okay, thanks. And I noticed, Richard, you did talk about the next stage in this latest drug being clinical trials. And Jenna wants to know how long it might take typically for a therapy to become available if a drug does turn out to be successful.
So we have a fourth PhD student now on this project. And the fourth PhD student is actually just coming to the end of his PhD. And what he's been doing is we learned fairly early on that OH14, although highly effective at doing its job in cells and in tumor models in mice, etc., when you have to perform a number of very strenuous and stringent assays to determine whether these compounds would be effective in humans. And some of the assays coming back was suggesting that it would probably get broken down inside the human body and probably wouldn't be as effective as we'd want it to be. So that was probably the death knell for OH14 itself. We still use it as a tool to show proof of principle. But what the PhD student has been doing now is looking at brothers and sisters of OH14, which will have exactly the same properties, very similar structures. So it's based off OH14, but we're looking for brothers and sisters now that will be a lot more stable in the human body. So he's actually now, right at the moment in the lab, back in the lab, he has approximately 50 additional relatives of OH14, which he's now testing in the laboratory. They are all stable forms of the drug, and it just needs one of those now to work as well as OH14 in the laboratory, and we're then at a stage to move forward. At that point, in 6 months' time. To get it into the clinic at that point will probably take us somewhere in the first in man clinical trials, which would just be toxicity trials, would probably take about 2 to 3 years before it went into a person for the first time.
Okay, so not the massively distant future.
No, that's fast.
Yeah.
Unfortunately, that is relatively speedy for a first in man molecule, and it comes back to that idea of repurposing drugs, which one of the original question I think was a great question, because if you can repurpose a combination of 2 existing agents that are already that have been used in the clinic, then you don't have this long lead-in time of having to test their toxicity, etc. You can get them in the patients quickly. So that's why we're so very keen on repurposing as well.
Absolutely. Rhiannon has a question. I don't know if it's too early to say this, but is it anticipated that this drug combination will be prioritised for patients once they're in remission to avoid issues of resistance?
Yes, so the issue of an acquired resistance or a sensitization is yet to be fully established. We think what we're seeing, certainly one of our publications from 2 years ago would suggest that actually C-flip is involved very much in acquired resistance, and we've demonstrated that estrogen receptor positive breast cancer patients who relapse on tamoxifen appear to have high levels of C-flip, and C-flip comes in and basically makes these they reduce the levels of C-flip and they become actually sensitive to this immune mediated killing. So actually the relationship between acquired resistance and the innate resistance in patients that have already seen paclitaxel, we don't fully understand yet. But the principle that we're working to at the moment is that this would be given upfront with the paclitaxel at the same time as paclitaxel to stop that accumulation of the cells to start with. Whether it could be used with patients in remission, we don't have any data on that at the moment. It would be fairly easy to follow up, but it would take us a couple of years, I think, to get to that sort of point. With the sort of experiment we would do before OH14 or one of its analogues got to trials, I think. So we would probably know before this drug was in trials.
Okay, so there's a question about what activates cancer stem cells. Stem cells lie dormant for a long time, especially in breast cancer. Can they act alone, or do they need a critical mass, a quorum sensing effect?
Yeah, very, very good question. I think Giusy and I could probably talk for hours on that one. Giusy, did you want, you're looking as if you're...
The thing I can say is that it is important also where these cells land. So they do leave the primary side, the breast, and then through the circulation, they might land elsewhere. For example, in the case of breast cancer, they might end up being in the bone or in the liver or unfortunately elsewhere. And I think that it is important to understand also the interaction between these cells but outside the breast. So the relation between breast cells and cells from other tissue, which might nurture them throughout time and somehow keep them happy and able to switch on again at some point and grow outside their actually normal environment.
I think the very simplest answer, which is a bit of an overgeneralization, but the simplest answer is yes. I think they work a lot better in pairs or in small groups of cells, and they don't have to be 2 stem cells. It can be a stem cell with an accompanying other cell, and they just help each other out.
But do they tend then, this is for me now, do they tend to clump together, or are they distributed as you were sort of, I mean, because you're saying they're rare.
Cells are very social creatures. They love to be in contact. In fact, actually, one of the fundamental biological processes, cellular processes, is a process of cell suicide. And you can induce a cell to commit suicide simply by removing it from its neighbors. It really dislikes being on its own. And this is where the cancer stem cells are unique, because they seem to be very resistant to that loneliness, and they're quite capable of circulating in the bloodstream on their own without dying and committing suicide. And that's one of the principal characteristics, right, Giusy, of a so-called cancer stem cell or circulating stem cell.
I see. Really interesting. So we've got to, I would say, 3 minutes left. Big question. Well, I don't know how big it is. Jack, in the pre-questions, asked how important is it to catch symptoms early? I imagine it's important. How important?
Oh, yes, definitely. Actually, I think this is important today more than ever to highlight these, even because we have to be reminded that cancer cells can start moving from the breast even before or whilst growing locally. So you don't have to wait for a big mass to form before actually cancer cells move out to the circulation and elsewhere. So that's why I think today's day when to need to be reminded to check ourselves, get checked. So no shame, no fear, and spread the word. I mean, tell your mom, your daughter, your sister, your friend to actually join any screening program available to them, because we know early detection and screening is the best way to actually fight against breast cancer.
So frankly, the earlier the better.
Definitely.
Yeah. Okay. Good. Well, I think we're pretty much at the end of our time now. So I've noticed, had a look at the list of attendees. I can see there's 70 people on the call. Thank all of you very much indeed for attending. I hope you found it as fascinating as I did, and that you learned as much as you possibly can. I know there'll be various levels of awareness and knowledge of the judging by the questions, which were excellent, by the way. Thank you so much for your engagement with that. And if we were able, everyone on the call, I'm sure, would be engaging in a big round of applause for our two presenters. But on behalf of everyone on the call, I'd like to thank you both very, very much indeed. And to everybody else, I hope we'll be able to see you back here next time. So thank you and good night, everybody.